Hi, Swati. It is 5 o'clock. We can start.
Thank you, Meghna. Good evening, good afternoon to all the participants joining from India and Europe. I already see many familiar names and glad you could join us for our AMR talks. We welcome you warmly to the second AMR awareness talk hosted by CCAMP as part of the CARBEX Global Accelerator Network. Before we start today's talk, let me take this opportunity to give you a quick overview of CCAMP. Meghna, if you could quickly just pull out the slides, please. So CCAMP is an initiative of the Department of Biotechnology, Ministry of Science and Technology, Government of India, and was created with the mandate to be an enabler of cutting-edge life science research and innovation in the country, and to act as a bridge between the Indian academic community and the industry. Next, please. So there are many verticals and the track of enabling entrepreneurship and enabling more innovations to come out from the academia to the industry and to the end user. There are various tracks that CCAMP currently enables. One is the obvious, by providing the seed money for many of the innovations to kickstart their journey at different levels of the entrepreneurship journey. And we've had support from partners like BIRAC, Niti Aayog, Ministry of Electronics, and also others, including many private partners. CCAMP also offers incubation facility where by many of the startups are given space and through mentorship programs as well as access to infrastructure, and through various accelerator programs. A few of them are mentioned as you see on the slide. Next, please. CCAMP has many infrastructure and enablements, as that is one of the core mandates of CCAMP. So on one side, you see high-end genomic facilities, proteomics facilities. We have mouse transgenic facilities, X-ray imaging facilities, and various others as kind of shortlisted and just given a quick overview here. And these are accessible to the innovators, and also the incubators have access to this freely, through which they are able to accelerate their own product development. Next, please. We take pride in having 200 plus strong innovations in our portfolio, and this spans across molecular technologies, across antimicrobial resistance, oncology, maternal and child health, across infectious diseases, non-communicable diseases, and medical devices and access and accessories. And this is again through various programs and through different processes. CCAMP onboards innovations, supports them in different ways, really customizing the support according to what is required. And this is what the portfolio holds currently, and it's growing year on year. Meghna, next please. So here you see a snapshot of where we are today. We have more than 250 startups which have been funded, incubated, and mentored by CCAMP. We have 20 plus life science healthcare products which are currently in the market, and about 770 crore plus INR follow-up funding has been secured by CCAMP startups. And there are currently 180 plus new patents which have been filed by CCAMP startups. So we take pride in what CCAMP has been able to accelerate in terms of the entrepreneurial ecosystem, the startups, in the various areas that we spoke and highlighted earlier. Next, please. And during the pandemic, it has been one of the most challenging times where CCAM has also kind of facilitated many of the innovations which it was incubating, also which it attracted during this time, and helped deploy it in the field so that it can be used to combat this very serious health crisis that the government, that the world is currently undergoing. And that is also an area where CCAM has made contribution, and you see the deployments are happening all across the country as of now. Next, please, Meghna. As of last month, we also launched the India AMR Innovation Hub. I'm sure many of you would have seen that come out in the media as well as on CCAM social media as well. And here it is basically the coming together of different partners. We already have a good set of partners. You see that in the lower panel, which come from the government, from international agencies, from the industry, and the startup ecosystem, as well as the academia and the research institute. The mandate is really to come together to enable solutions in AMR that can be fast-tracked. And this is just launched, and we are hoping to work across human antimicrobial resistance, also taking the One Health approach, where we are looking at animal health, human health, and environmental factors that contribute to AMR. And also the third angle being AMR in neglected tropical diseases. So this would work across diagnostics, therapeutics, stewardship, awareness, and so forth. And coming to today's talk is one such awareness initiative that we have taken. And through these series of AMR talks and webinars, we hope to take that important first step towards combating AMR and holding more awareness talks. And that is the best defense against AMR is how to prevent it. And the more the people, the institutes, the organizations are aware of the threat, the more impactful any intervention introduced henceforth would be. So today's talk, we are glad to host. It's on IHMA. The title of the talk is Your Global Antimicrobial Partner for Fighting Against AMR. And we are happy to welcome our guest speaker today, Dr. Ian Morrissey. He's the Chief Scientific and Development Officer, IHMA Europe. Dr. Ian received his PhD from the School of Pharmacy, University of London in 1990, and an executive MBA from Imperial College Management School, University of London. He has served as CEO at GR Micro, which was later acquired by Quotient Bioresearch, and he continued there as a director of microbiology until 2012. Since 1987, he has been actively engaged with the pharma industry to support development in new antimicrobials, particularly preclinical and surveillance studies. Dr. Ian has also authored more than 250 publications in AMR and associated areas. He's a fellow of the Royal Society of Medicine and member of various reputed committees like BSAC, Standing Committee on Antimicrobial Drug Discovery and Development, and so forth. Dr. Ian, it's our pleasure to welcome you to this forum today.
Thank you very much. Thank you very much, Swati, for your kind introduction. It's an absolute pleasure to be able to present here today. I'm very happy to say that I had the honor of visiting your facilities a year or so, or a couple of years now actually, back, and I was very impressed with the setup. So I think people doing research in this area in India are very lucky to have you there on their doorstep as a resource for our fight, as we're calling it, against antimicrobial resistance. So let me
Thank you, Dr. Ian. Thank you for those kind words. And yes, this is something, AMR is something that we all need to, prepare ourselves with. And that is what we're hoping your talk would also enable many of our participants working in this area would benefit from. I now invite you to share with our audience about IHMA and how it is enabling researchers, innovators and institutions develop solutions against AMR. So over to you, Dr. Ian.
Great, thank you. It's my pleasure. So I have a few slides on IHMA, and hopefully we'll have some time for some questions as well afterwards. This is our mission statement. So IHMA is a very focused company offering microbiology services for companies wanting to develop new antimicrobial agents or maybe vaccines, and also diagnostics in the area of antimicrobial resistance. And we have expertise offering laboratory services and also consultancy experience with various members of the company having many years of experience working in this area. IHMA is a global company which originated in the early 1990s in the USA, a place called Schaumburg, which is near Chicago, Illinois, and started off actually as a consultancy company and then grew into a fully supported microbiology laboratory. And then a few years after then, in the early 2000s, IHMA Europe was set up in Switzerland, which again has a whole array of laboratories offering microbiology testing. In Switzerland, we have around about 25 or so staff. In the US, it's a little bit more than that, maybe 100, 120, something like that. But we offer the same microbiology services within those laboratories. And I will talk a little bit more about that in a moment. Moving up to more recent times, but actually probably the most relevant for the audience here is in 2018, we set up an operation in India. And I'm very pleased that we have our CEO in India on the call today, Dr. Tarun Mathur, who is heading up our operations. We do not at the moment have a laboratory in India, but maybe it's something we will set up sometime in the not too distant future. But at the moment, we're offering support for companies within India in particular, but also global companies that have an interest in studies and an interest in bacterial isolates and fungal isolates that come from India. And again, I'll talk a little bit more about that in a moment. And then finally, more recently, we set up a laboratory operation in China. So we have a relatively small laboratory in China that carries out most of the same microbiology activities that we can perform in Switzerland and in the USA. But at the moment, it's still growing. So we don't do absolutely everything in China, but we do a reasonable amount of work to support companies that need data from isolates originating from China. So when we talk about tackling antimicrobial resistance, what do we really mean? Well, we have companies that are doing things on the left there in blue. They're looking at new strategies to fight antimicrobial resistance. And as we know, and I think everybody is aware, that the big problem with anti-infective drugs is that the bacteria or the virus or the fungus manages to develop resistance by mutation or acquisition of resistance genes that enables perfectly good antibiotics to become redundant and useless because resistance has developed. So companies need to come up with new strategies, new ways of maybe finding new chemicals, maybe new ideas looking at bacteriophage, which isn't really a new idea, but it's something that hasn't really been looked at in as full a way as perhaps small molecules have. Also looking at maybe peptides as an alternative for antimicrobial compounds, or looking at vaccines, etc. And also within these strategies are also ways of detecting resistant. So there are numbers of companies in the diagnostics area that are developing tests that can enable not only the detection of resistance, but maybe also to predict antimicrobial resistance. So I think there's lots of clever people within biotech companies, within universities, within larger pharma that are coming up with new strategies. But also in relation to that is knowledge. We need to be able to understand, what are the problems with antimicrobial resistance, because this is something that doesn't stand still. So I was describing a moment ago, you have a perfectly good drug one day, and then perhaps a few years later, it's no longer of any use. But we only understand the extent of that problem by collective knowledge, by looking at the organisms that cause infections, measuring their susceptibility, so we can get a very good picture of how that relates to now and how we might predict might happen in the future. And this is really where IHMA comes in, if you like, as a link, a sort of partnership between the knowledge that's there and also the companies that have new strategies. And one of the main ways that we do that is through antimicrobial resistance surveillance studies. And by doing these studies, we can partner very well by offering the knowledge from these studies to those companies that are developing new strategies. And also our surveillance studies are a very good resource for these companies to do smaller early studies, but very much focused on resistant organisms. And the example that I always use is a number of companies in recent years have developed compounds that they believe will target beta-lactamases, in particular metallo-beta-lactamases, but other beta-lactamases as well. But a number of these resistance mechanisms are actually quite rare, and it's very difficult to find a collection of these strains in most places. But we collect a large number of isolates every year, and I will talk about that in a moment. And within this collection, we can cherry pick those organisms that are of interest. So if a company is at a very early stage, or perhaps screening their molecules to see which is the best beta-lactamase inhibitor we can find, we can put together relatively small panels of organisms that can be used as part of the screening process. So although we do really large surveillance studies, we also do very small early stage studies as well, which is very important to really kickstart companies so that they can get as much valuable information as possible at the lowest cost possible. Because we understand that the market for anti-infectives, everybody talks about the economics behind it, the model is broken, etc. We know that companies do not have a lot of money, certainly at the beginning, to spend on their research programs. And we understand that they need these data to get grants. So most companies are getting grants, whether it's from Carbex, which is more relevant for today, but also with other grant giving bodies. They need some kind of data to support their concept. So we can offer very small but very focused studies to help generate that data to kickstart companies to then have a nice set of information to present to those organizations that are providing funding for antimicrobial resistant studies or new molecules. And this is a quick picture of the world just to give you an idea of where we operate, which is pretty much everywhere, as you can see. Probably our weak spot, if I was to focus on that, is Africa. We have mainly sites in South Africa and then sites in Northern Africa, but not so many in between. And that's pretty much related to the infrastructure in Africa, but also the companies that are engaging with us to do their surveillance studies are particularly interested in those commercial markets. So they're not necessarily interested in surveillance for academic sense. They're interested in surveillance for the markets where they believe they will sell their compounds. Although this dynamic is beginning to change. So with Carbex, they have a particular interest in lower and middle income countries where surveillance of resistance is important. And I think these kind of organizations will be focusing on those kind of countries that are missing here in our map. And we're working with these organizations to see how we can improve our surveillance to cover these less well known regions of the world. So this really, I won't go through all of these countries. It's just really to illustrate that our surveillance is pretty extensive. But I will focus a little bit on India, because obviously that's very pertinent to the discussions today. So we have a very nice surveillance network in India, which has been enhanced since Tarun has joined IHMA and set up IHMA India. So we have 14 sites within the country that are active in various surveillance programs, and you can see those numbered in blue in the map on the right-hand side. And so it covers quite well the regions within India. Not perfect, of course, but does a pretty good job. And we have a number of other hospitals that are on standby. So Tarun has a lot of contacts within India. He's worked previously in the pharmaceutical industry in India with Ranbaxy and Daiichi in particular. So he knows a lot of people, and that's very useful. So we're in a position to expand this surveillance as and when it might be required. So these 14 sites are those that we're actively doing studies. There are others there waiting in the wings to participate, and very keen to participate in our surveillance should the opportunity arise. And the surveillance is pretty extensive, looking at most, if not all, of the gram-positive and gram-negative isolates that you would perhaps be interested in, also including fastidious organisms like Streptococcus pneumoniae, Haemophilus influenzae, and also trickier organisms like Neisseria gonorrhea. And I say tricky because, well, they are quite tricky to grow, but more relevant that a lot of hospitals do not actually culture this organism anymore. They rely on ELISA or PCR type technologies to detect an infection rather than trying to grow the organism. But we have a network of laboratories that can help us with these kind of isolates. And we are actively doing research for some companies that are developing anti-gonococcal drugs. And also, we do surveillance in the fungal arena. We are best known for antibacterial work, but we also do surveillance in mycology too. And this is a growing area for IHMA. And the last point is probably the most important point, is we have authorization from the Indian government to export isolates from India to our US and our Swiss laboratories. And this is very important because we can now include India isolates in parallel testing with isolates from other parts of the world. As all of you here, I am sure, aware that there were issues, shall I say, with the export of isolates from India, and there were various quite harsh restrictions put in by the Indian government, which really made it very difficult for companies to export isolates out of India. And that meant that in the past we had to rely on working with partner laboratories within India, which is not really a problem directly per se, but is not ideal, because it's always better to perform our studies all together within one laboratory, because there is inherent variability from lab to lab. So the more that you can do in a centralized lab, the more robust your data will be. And Tarun again was very, very active in this to apply to the Indian government and explain the research that we were doing, the importance of this research to India and also the rest of the world. And by doing so, we were unable to get permission to export these isolates. And the way we work with this is we have a central partner laboratory indicated here with the star, and we work with them. They will hold our isolates until we're ready to have them shipped. So we have a repository of India isolates that are maintained within India. So this is a resource which we're more than happy to provide isolates to other companies if they wish to use those as part of their research. So that may be something that some people here in the audience are interested in. We will be more than happy to provide those isolates to companies should they wish to do their own research. And of course, we can test those isolates with any new molecules, etc., provided by other companies as well. And we have worked with a few companies within India to do this type of work, mainly within our Swiss laboratory. So we will get molecules from India sent to our Swiss lab, and then we will test India isolates as well as other isolates from around the world, as required for the study. So really, I guess one of the take-home points of my presentation is really the core of IHMA's offering and uniqueness is our collection of isolates. And we have well over 1 million isolates that have been characterized phenotypically. So we know the resistance that these isolates might have. So as I was talking about earlier, we can cherry pick, put very bespoke panels together of organisms with certain resistance and other determinants that are useful for companies, particularly at an early stage. And we're continuously refreshing this. Anything from 100 to 150,000 or so isolates are collected every year. And that's obviously very important because resistance changes quite rapidly. So we cannot rely on isolates that are 10, 5, 2, or sometimes even 1 year old. We really need to keep on refreshing our collection. And here's a couple of pictures there of our freezers. Really, a major resource for us is keeping these organisms safe and sound for other people to then use for their research, or for us to do the research on their behalf. So we have continuous monitoring of these freezers. They're the most precious things within our collection. So there are people who are on standby just in case there should be some kind of alarm, and they are automatically sent a text and an email to inform them that there's potentially a problem, and they then have to go rushing into the laboratory and check that everything is okay. So this is very, very vital and very, very important for us. So what does IHMA actually do? Well, we support all of the phases of antimicrobial drug development, and we support them by performing the in vitro testing. So we don't do in vivo animal modeling, we don't do toxicology. We are very specialized and very focused on in vitro microbiology. And that can range from drug discovery through to clinical development. And we can also help companies with their regulatory submissions and a little bit for commercialization. This is more for commercialization of diagnostic devices, particularly devices for susceptibility testing. And this is a major area for us, for companies that are maybe developing discs, are developing gradient strips or automated systems. We can support those companies with the commercialization of those devices within hospitals. This is a very, very busy slide, but I wanted to put everything together on one slide to again emphasize the point that we can support companies from the early drug discovery through to their later larger research and development studies, including clinical phases, to the approval and also the commercialization of diagnostic devices. So going from left to right, sort of preclinical testing, we perform a lot of that for companies at various stages. Sometimes it can be screening of large numbers of molecules. We don't do robotic sort of high throughput screening of thousands and thousands of molecules, but we can work with, say, tens of molecules if needs be. Most companies, they tend to have maybe half a dozen or so compounds within a class that they're particularly interested in, and we usually focus on that, but we can do larger panels if required. So these can be relatively small with regard to numbers of isolates, but can be very focused on isolates that are of particular interest to the company in question. And the main test that we do is the standards MIC determinations, so minimum inhibitory concentration determinations. But of course, there's a whole raft of other microbiology studies that need to be done as a drug is being developed, or as a company is deciding what will be their lead molecule and what might be their backup molecules. So these are things like MBC, so minimum bactericidal concentration. So an early, relatively basic test to see does your drug kill or not. but then proper killing studies like time kill studies, checkerboard studies looking at interactions between the new drug and other antibiotics. This can be for a direct purpose because you're expecting to have a synergistic, a sort of a positive effect of the combination. So again, my example of beta-lactamase inhibitor, that type of checkerboard is kind of obvious to see that you expect the beta-lactamase inhibitor to protect the beta-lactam drug. But also checkerboard studies are important to be sure that there isn't an interaction with other antibiotics that are currently available. So a number of the problem organisms these days are those that cause infections in seriously ill people that may be in intensive care, that may require to be given a number of antibiotics all at the same time. So we would do checkerboard studies to combine the new drug with existing drugs, just to double check that there isn't an obvious problem with antagonism between the new drug and other drugs. And that's very important to know that. And other things like PAE, post-antibiotic effect, is also of interest sometimes, although I must say it's not so popular as it used to be. A few years ago, everybody was interested in post-antibiotic effect. I still think it has a role to play. It can be useful to help decide what type of dosing regimen is required. So the post-antibiotic effect is basically the effect that continues after the drug has been expelled from the body. So there may still be an interaction with the organisms, which would mean, although the drug isn't there in the blood, it's still having an effect against the organisms. So perhaps you can spread out the dosing. It's obviously not full pharmacokinetic type studies, but it's part of the jigsaw to decide, what would be a sensible dosing regimen, particularly in early animal model studies where it can be fine-tuned before ultimately you get into man. Also, testing conditions are quite important. A number of molecules these days are a little bit different. So the new strategies, the new ideas that I was talking about earlier, often involve compounds that are quite different. Things like antimicrobial peptides, things like phage, that don't work very well under standard testing conditions. So sometimes the method has to be changed to enable the appropriate testing to be done. And we look at that kind of thing and help companies decide, what is the best testing method to be used. And then a little bit later on, what's very important is to understand how this testing might work and understand how quality control might work as well. So ultimately, when you have a drug that's on the market, it's being used in hospitals, the hospitals will need to test those drugs. So you need appropriate quality control to ensure that the tests that they're doing are robust. So we also help companies, particularly working with the CLSI in the US and also UCAST in Europe, to enable these quality control strains to be set up, and also for the reference ranges for these strains to be set. So there's lots of these little more less academic research, if you like, and more procedural research that is still very, very important. And a lot of companies forget about that. They're very excited about the breakthrough novel ideas and the fantastic results that come from that, but forget about the fact that the humble clinical laboratory also has to test this new drug, and they need to understand how they do the testing and what quality control is necessary and what reference ranges are acceptable or not. So this kind of thing is a bit dull and boring, but is vitally important and is really essential to help a new drug be used. We also support clinical trials. We're not a clinical research organization, but we will work with these CROs to act as a central microbiology reference laboratory. So we have a lot of experience in the microbiology side of it that hopefully I've demonstrated already, but also we have a lot of experience in logistics, so sending the kits to the various hospitals within the clinical trial. And also we have a lot of experience with data collection and data analysis, which again is a vital part of the clinical study. So we work hand in hand with the CRO to actually make sure that the hospitals are doing the right things, they have the right kits, that we receive either the clinical specimens or the clinical isolates. We can do the testing and present those results back to the CRO for part of their later analysis. And there's lots of things that are required for this. And we can support all of these areas as sort of listed there. I won't read them out. Then the last point I've kind of touched upon already is our resistance surveillance. So this is an important, it's actually a mandatory activity that is required for any new molecule. So as you get towards maybe the later stages of development, maybe sort of late phase 2 and early phase 3, at that point, there should be robust, relatively large surveillance already in place that really augments the data from the clinical studies. So the idea of these clinical surveillance studies are to collect isolates from similar countries where clinical studies are being performed, but in much larger numbers than are possible in a clinical study. As you can imagine, a clinical study is an expensive thing to do. You have to recruit patients, you have to monitor the patients. It's not something that comes at a low cost. So the way this is augmented is by using surveillance studies where you don't need to recruit the patient, you don't need to do all the monitoring, you just need to collect isolates from relevant patients. And these are the hospitals in the network that I was showing you earlier. They send these isolates to us for us to then include in these mandatory surveillance studies. So that's the primary purpose of these studies. And as I say, it's essential and every company has to do these type of studies. But what is useful is that these isolates that we pick from our surveillance can also be fed back into other companies' research at an early stage. So we can put panels together of organisms that may be of interest to them. So as you can imagine, if you're collecting 100, 150,000 organisms every year, you're going to pick up some very interesting organisms there that are quite rare, that perhaps any one hospital around the world is not likely to have these isolates. So it's a numbers game, if you like. We collect so many, we're bound to have isolates that are particularly rare, but very interesting and very important for companies to test as part of their sort of due diligence and their proof of concept type studies at the early stage of their development. And then the last point here on the slide is something that we call adjunctive services, and something that we do which is more for companies that are developing antimicrobial susceptibility testing devices, but we have something that we call AVETs. And what this is we provide these new devices to clinical laboratories around the world. And why we do this is because there is always a gap between a new drug being approved, whether it's in the US, Europe, India, or elsewhere. There's always a gap between getting this approval and then the companies that actually are making the susceptibility testing devices being allowed to sell those devices. And the reason for that is a company will not get its official clinical breakpoints until right at the very end, when the clinical data have been looked at, when the susceptibility data has been looked at, when the PK/PD data has been looked at. So there's a lot of number crunching that goes on that ultimately FDA or EMA or the Indian government or whoever says, okay, these are your breakpoints. Only once those breakpoints are provided can the companies that make the testing devices do their final studies to then get this device approved for official clinical use. And that can quite often take 6 months, 12 months, sometimes even longer than that. So you have this gap where a company is allowed to sell their drug, but the clinical laboratories cannot test the drug because there aren't official devices that can be used. So we bridge that gap by providing these devices to the hospitals for their own research and development. So they cannot use these for clinical testing, but what they can do is test their isolates and see what does the activity look like. And this is very useful for not only the susceptibility testing device companies, but also the pharmaceutical companies that is selling the drug in the first place, because this gives first-hand options for these labs to test the new drug, which otherwise they wouldn't be able to do. So this enables these drugs to be fast-tracked onto the formularies of hospitals, because generally hospital pharmacies in particular that decide, are we going to have this new drug or not, are always interested in knowing, well, how does this drug work against the isolates that we're seeing on a day-to-day basis? So by us providing these devices, the hospitals can, or the microbiology lab in the hospital can show the data to their formulary team and say, look, this looks really good. It looks much better than other drugs. So when we are able to use it, we should be able to do that, immediately. And that's a very useful activity that we do. And also we get involved in a lot of publications, a lot of posters with obviously abstracts and posters presented at national and international meetings, and also film manuscripts as well. And we recognize the rigor that's involved in somebody presenting a dossier for regulatory approval. So all of the reports that we produce from our microbiology studies are in a format that's of an acceptable quality to global regulatory authorities. They will look at the reports and they will see it's of the appropriate standard required for them to recognize the data, which again is very important. So I think I'll stop now just really with a summary that we're a pretty unique lab in the sense that we have facilities within 3 continents and very much focused on microbiology and microbiology alone. One thing I should say, which actually I didn't point out before, is we do not work in virology. We only work in bacteriology and mycology. And we have 3 active laboratories, and I hope one day in the future we will have a 4th active laboratory in India. At the moment, we only have 3 laboratories at US, Switzerland, and China. These laboratories are accredited by the College of American Pathologists. What this means in particular is we are, that enables us to perform clinical studies. So without this level of accreditation, we would not be able to support clinical studies as a central reference laboratory. And really, if I was talking to any one of you at a conference and we were in the hotel in the elevator and I was giving my elevator pitch, I would say in a nutshell, we support all of the in vitro microbiology studies that are required to develop a new anti-infective drug. And importantly, we have an isolate repository, including India isolates, that are available for IHMA to do studies on, but also we're more than happy to provide some of these isolates to companies if they want to do some of their own research on those isolates. And I will stop there. Thank you very much, everybody, for listening, and I'm happy to take any questions. I can see there's lots of things in the chat, but I wasn't able to read them as I was talking, but I'm sure Swati will help out with that. Thank you very much.
Thank you, Dr. Ian. Yes, the chat has been buzzing throughout your talk, and I see Tarun has been kind to answer many of these questions. But
thank you, Tarun.
It's a pleasure. But I think we can take any questions now onwards because Ian is here, and I think we can take an advantage of his presence.
Yes, so I'll take a few questions from the chat, and one was whether IHMA provides facility of clinical trials required for approval of products in the US and the Europe.
Yes, so we don't perform clinical trials ourselves, so we're not a CRO, but we perform the microbiology that's required as a central reference laboratory. So what that means more specifically is if you're doing a trial for an anti-infective, quite often there will be local testing by the hospitals enrolled within the clinical study. But as I was describing earlier, you can get a fair amount of variability from hospital to hospital. So what tends to happen is the clinical trials are designed in such a way that there is a central reference laboratory that will perform confirmatory testing of the identity of the pathogens that have been isolated as part of the trial, but also the susceptibility of those isolates to not only the new drug, but also other comparators that are clinically relevant. So there's 2 parts to that. One is obviously to confirm the activity of the new drug, but secondly, also to have a nice panel of other drugs to really put the activity into context with these comparators. And quite often the clinical laboratories do not have the facilities to do full-blown MIC testing. So for the clinical studies, the testing has to be using a recognized reference method, which is normally for most isolates, either the CLSI or UCAST broth microdilution methodology. And quite a lot of hospitals don't perform these types of tests routinely. They often rely on automated systems or discs or sometimes gradient strips, so that it's not so easy for them to perform the testing. So it's not just the variability, it's also quite often it isn't even possible to do this type of testing locally. So we come in as the reference lab to confirm that activity. And then also as an adjunct to that, we also will perform molecular studies as well. So sometimes you may get interesting organisms. Perhaps an organism has caused a breakthrough infection during the trial and they want to know, is this something to do with resistance development? Or there may be some other factor. Maybe it looks like there could be a clone causing a problem within a hospital. We can then do the molecular studies to see, does it appear to be the same clone causing the infections, or is it a number of unique separate organisms? So those types of questions are often required as a secondary part of the clinical trial that wasn't necessarily planned on day one, but become important as the trial progresses. So that's how we get involved in these studies, and the data that we present are of a standard that would be part of the regulatory requirements, but we don't run the studies ourselves. We rely on CROs. We have a very close relationship with a CRO called MedPace, but we're also free to work for any other CRO that a drug company may wish to work with. But we work mainly with MedPace.
Thanks, Dr. Ian. So a more general question, any entity with interested in early stage screening or who needs support for device development and testing, what is the general route they would take to approach you, and what is it that you require from them before, to have a successful relationship and a successful initiation of testing studies with them? So
okay,
very broadly and very generally kind of point.
Okay, sure. I mean, I think you used a very, very key phrase there in the word relationship. We very much feel that we like to work as a partner with companies, not in the true full legal sense as a partner, but really as somebody that not just does studies for you, but also, we're very happy to engage and discuss what might be required. So for us at an early stage, the most important starting point is to know what data are available already. So what studies have been done, so that we know at what stage within the continuum we need to pick up from. So hopefully you would have seen from my slide, we start from really small studies and we work our way up with sort of larger studies as we get confident with what is going to be the lead molecule, et cetera, et cetera. Obviously in parallel, there's toxicology studies and other various non-microbiology studies that need to be done. But if we know, for example, the company has already done some early screening and they have some nice data that demonstrates, this is the lead molecule, we don't need to reinvent the wheel. So we don't want to be doing studies for people and charging people money to do studies when they don't need to do it. So if companies can share the data, we can say, okay, that's good, maybe something is missing. So really the key for us is an open relationship with the company, so we can really do our best to advise them as to the next steps that they need to proceed with. And then once we make a start, it's kind of basic things like, what are the properties of the molecule? Is there anything particularly problematical about preparing solutions? And all sort of those technical kind of details are also very important. Once we set the ball rolling with the process, then obviously other questions arise as we sort of go along. But generally, that sort of information is really important. What do you have so far? Particularly if there's a molecule, I mentioned it a couple of times, in molecules that have unusual testing requirements. It's very important that we challenge that requirement very, very thoroughly. So people like CLSI and UCAST that are sort of international organizations that have an overall responsibility for testing, they will not just simply accept somebody saying, oh, we need to do this, it's a bit different. there has to be robust data to support that. And sometimes we work with companies and they say, oh, we have to use this test medium. And when you look at it very carefully, there's actually, the reason is if they change the method a little bit, their MICs, the currency of testing, if you like, are nice and low in their minds. And they like that. Everybody likes that. The lower the MIC, the better. But that's actually not so important. If you use the standard testing and your MICs might be higher, that really doesn't matter. The most important thing is that whatever you're measuring in the test tube reflects your clinical outcome. So places like the CLSI will not just accept somebody saying, oh, well, we want this method because the MICs are lower. They will only accept a new method if you can demonstrate that there is something wrong with the standing method. So this type of discussion, you know, companies need help with that, and we're more than happy to help them. So again, it's having an open relationship, providing us with as much information as possible so that we can really advise companies. Because what I would hate to do would be for us to run a lot of studies for a company using a new test, for then the CLSI or UCAS to turn around and say, these data are useless. You know, we can't support your request for a new method, because that would be, you know, a terrible outcome for everybody. So openness is important.
Thanks, Ian, for answering that question. I'm sure many of the early stage startups and would be interested to know that. And thanks for clearing that. I'll take a question from Professor V.S. Chauhan, and thank you, Professor Chauhan. It's very nice to have you on this forum. His question is, will you support and or collaborate for early preclinical development of promising antimicrobial agents? He says we already have in vitro and in vivo efficacy data with MDR and XDR bacterial strains.
Absolutely, yeah. I mean, we're more than happy to help companies move to the next stage. I guess one thing to say, which is quite important, is we do not get involved in co-development. So we work, as I say, as a partnership, but purely as a financial transaction. So we do a study for you, and that's we then have, require a payment for that study. So we don't say, okay, give us 5% equity in your development plans and we will do those studies for you at no charge. So we work really at a hands-off or arm's length relationship as far as the commercialization side of things. We have been asked many times over the years whether we're interested in having a financial interest in companies, and we decided that there's too much of a conflict of interest for us. If we're working with company A and all the other companies in the world know that, oh, IHMA are working very closely with this company, I always fear that they will think, okay, there's a bit of a bias against that company, and perhaps when we need to do a study done, if this company A also wants a study done at the same time, they will get priority. Maybe IHMA will, leak some of our information to that company A. Of course, that would not happen. Confidentiality is vitally important for us, but I understand that companies would be concerned about that. So we've always steered clear of financial partnerships and sort of collaborations in that way. But certainly for any type of study, we're more than happy to work with companies and do the studies for them that way, but not as having a share in their development. We also are quite happy to, and we have done them from time to time, help companies with maybe their grant applications and things like that. Perhaps not to fully write their applications for them, but we're always more than happy to review any applications, perhaps suggest what microbiology studies might be required and that kind of thing. We don't charge anything for that sort of consultancy. Obviously, we hope that the grant application will be successful and the company will come back to us and do those studies with us. But that's obviously not guaranteed, but we're more than happy to help companies put their thoughts on paper and help them design the program that they're hoping to get funded.
So that was going to be my next question, actually, when you had mentioned when you mentioned earlier that there is a kind of a relationship that IHMA builds with the different startups in terms of a partnership. And so my thought process that was going on is, if somebody is interested for insights while writing a grant, how IHMA gets involved, and does it get involved as a collaborator or simply as somebody who's guiding on that particular aspect of investigation. So I think
we are very happy to be a formal collaborator in a grant application as well. The only stipulation that we have is that the funding for industrial collaborators, shall I say, is full funding. So some grant applications sort of under research and development are set up such that you get, let's say, 50% of your requirement by the grant funder, and then the remaining 50% has to be covered by the industrial partner. For us at IHMA, that's not really suitable for us, because we're not developing a product. We're just doing the tests to support the company that is developing a product. So if it's only partial funding, then we wouldn't be able to be a partner in that type of application. But if it's something where it would be full funding, then we're more than happy to be a formal part of that application.
Thanks. I saw one question which asked about the charges, and what would be the charges or the fee for IHMA for different purposes. Would you like to take that, or is that something that comes out of after discussion and really scoping out the partnership, and till what extent IHMA is going to be involved?
Yeah, I mean, it's very much, the devil is in the detail type answer. It really can vary quite a bit depending on what is required. Because of that, we don't tend to have a price list. Obviously, once we start doing discussions, we would have a confidentiality agreement in place, a two-way one. So obviously, any information that a company provides us on their molecule, which I was talking about earlier, but also any pricing that we provide to them is covered by confidentiality. So I don't really want to go into too much detail about that. But I can say that the smaller sort of early screening type studies would be around about sort of $3,000, $4,000, $5,000, something like that. For us, we think that there's an optimum size of study that a company should do. It's not very useful to do really, really small studies because they don't really tell you a lot. So we tend to advise companies to have a reasonable size study so that they can get a decent amount of information in one go. Because actually, it's more cost effective to do one study with 100 isolates than it is to do 10 studies with 10 isolates. So they don't tend to be incredibly small studies, and usually would be maybe minimum size of 50 isolates. That kind of size is what we do. So approximately $100 per isolate, something like that, which could be basically a screen for, say, half a dozen new molecules, or it could be a screen that just includes one new molecule and then existing molecules. So that's a kind of ballpark of the sort of starting early screen type study. And obviously, as these studies get more complex, either in the number of isolates or in the techniques like kill curves or resistance studies, then obviously they become more detailed and more expensive as we go along. But just to give you an idea, a sort of 3,000 or 4,000 or 5,000, something like that, is an entry level study.
Thanks, Dr. Ian. So I'm seeing that we're totally out of time, but there are still a lot of questions which we could not take up during the session. And they are more, in terms of very specific questions in terms of whether IHMA deals in escape pathogens or certain resistant profiles of certain bacteria amongst UTI bacteria. What is this, spectrum of isolates that are there in terms of also for E. coli, for MTB. I see Tarun has answered a few questions, but I would request Meghna to also take these questions, which we can pass on to Tarun and see how these can be answered.
Great, thank you. I mean, we pretty much cover everything with regard to
Yes.
pathogens and infection types. We don't work with TB, so we don't have biosafety level 3 facilities. But apart from that, we collect pretty much everything as far as pathogen scope is concerned. And obviously within those sets of isolates, we have most resistance that has been heard of. So we have pretty much everything covered.
Yeah, well, just to add on what Ian said, Swati, I mean, we'll be more than happy to take a few questions. In fact, some of the members who have attended today's call, they have a specific questions regarding the certain number of isolates what we have, a particular resistant type. So, I mean, it's being challenging to answer every question right now in terms of the numbers. So, I mean, I will request you that if you can share our email address, although I have posted my email address along with the IHMA website address in today's in the chat box. But somebody is having a specific question, please do share with us and we will certainly respond to them accordingly. Thank you.
Thanks, Tarun.
So I hope, Meghna, you have taken a, you've recorded this chat, and maybe we would have the email IDs because most of them are registered for this particular talk, so we can possibly pass it on to Tarun, and he could revert on this individually as well.
Yes, I have noted down the questions, as well as I'll pass on the email ID which Tarun has provided.
Thank you.
Yes, thank you.
Thanks. So before we close, can I request all of our participants and the organizing team as well to please switch on their video for a screenshot, a tradition that we follow, that we try to capture whoever has been attending. Good to see you, Professor Chauhan. And Meghna, do let us know when you are done.
If I can request a little more switching on cameras, please.
Everybody's very shy today.
So maybe you can capture the ones who are not camera shy, Meghna.
Yes. Yes. Thank you, everyone.
Yeah, thanks. We thank Dr. Ian Morse for his time and for sharing information on IHMA and how it can enable the AMR innovator in their journey. We hope that has made many things clear for our participants today. We also thank our participants for active participation, keeping the chat buzzing. Those whom we could not take up, as we said, we'll try to pass on the question as well as the email IDs to Tarun to take it up and answer, one-on-one, because they are very important and we would like that you should be supported and get your answer. Also, for future such talks, please keep checking CCAMP social media and announcement and on our activities on AMR. So thanks, Dr. Ian, once again. Thanks, Tarun, for enabling this discussion. Thanks, Meghna, for handling the back end. And thanks, everybody, for joining in. Bye, stay safe, and season's greetings and happy holidays to you.
Can I just say that this was an excellent talk. Thank you very much for giving the talk and wishing you, all of you, a happy new year.
Thank you, sir. Thank you very much.
Thank you. My pleasure. Bye, everyone.
Thank you.
Bye.
Bye-bye.
Thank you, everyone. Bye-bye.
Bye.